we have discussed about the rhizosphere, in which we have seen we have discussed about the signaling molecules also. The signaling molecules, which are diverse type of molecules are there, chemical compounds are there, which are present in the rhizosphere, and they have diverse function also. They act as a signaling molecule for different kind of microorganisms. Okay, so when the concentration are increased or the microbes identify these chemical compounds in a certain specific concentration, then they activate also. Their genes are also activated for their symbiosis or for their interactions or for their entry also. Okay, so these are important for the rhizosphere effect, as well as some symbiosis process which takes place into the soil plant systems.
Excuse me, sir. Sir, could you explain the concept of RS ratio once again?
Okay, the concept of RS ratio, R means rhizosphere and soil is for soil from the non-rhizospheric soil. Okay, is it clear?
Sir, but when we consider the effects of the RS ratio greater than 1, equal to 1, or less than 1, so we are talking in terms of the area of the root
Population.
expansion, or we are talking of microbial population?
No, no. We are talking in terms of the microbial population in the rhizosphere. Okay, so for
Yes,
this
sir.
purpose, one practical is there, and in which you can identify the rhizospheric effect. For this purpose, you have to quantify the microbes in the rhizosphere. Okay, so for this purpose, the practical approach is you have to uproot the plant from the fields. One plant you have to uproot from the field, and another from the barren soil, barren field you have to collect soil. Those soil are analyzed for the microbial population. Okay. Some network issue is there.
Excuse me, sir.
Yes, network issue is there. I describe you. हाँ डॉक्टर साहब हाँ भेज देंगे डॉक्टर साहब अभी क्लास ले रहे हैं उसके बाद भेजते हैं Okay, so we are discussing about the RS ratio. R for the rhizosphere and S for the soil from the non-rhizospheric soil. Okay, so when plants are uprooted from the field, rhizospheric soil are collected. Rhizospheric soil are the soil which are there on the root surface. Okay, so if those soils are enumerated for the microbial population, then the microbial population count in both soils are entirely different. In the rhizospheric soil, there will be huge populations of microorganisms are there, whereas in non-rhizospheric soil, the microbial populations are very less. And RS ratio is the ratio of both microbial population only. Okay, so if population is more than one, then rhizospheric effect is there. Okay, so rhizospheric effect is there, and if population is equal, then the rhizospheric effect is negligible, and if it is less than 1, then the rhizospheric effect is negative. Rhizospheric effect is there. Okay, now it is clear.
Yes, sir.
Excuse me, sir.
Yes.
Sir, how the foliar application of nutrients will alter the rhizospheric microflora?
Okay. So the last class we have discussed also regarding the foliar application of nutrient. So when you apply foliar nutrient on the foliage of the plants, there will be great change in the activity of the plants will be there. Physiology of the plants is affected. Photosynthesis of plants will be there, and during the photosynthesis, uh, carbohydrate and other six carbon compounds are synthesized, which have to combine with the nutrients to formulate different type of materials. Okay, so during this process, the root exudation process will be enhanced because there is physiological change will be takes place because nutrients are coming from the leaves as compared to the nutrients which are coming from the soil. Okay, so a lot of changes takes place at that time into the rhizosphere also, and exudation process is also takes place into the soil. So in that case also the rhizospheric effect is seen, and there will be alterations of the microbial population in the rhizosphere. है ना? तुम लोग के पास सुब्बाराव की बुक है माइक्रोबायोलॉजी की?
No.
सुब्बाराव की बुक में इसमें डिटेल में दिया हुआ है, तो अगले क्लास में मैं डिटेल में केमिकल कंपाउंड्स के बारे में मैं बता दूंगा कि कौन सा केमिकल कंपाउंड्स उस समय एक्सक्रीट होता है, एक्सक्रेशंस होते हैं, है ना?
Okay.
थोड़ा
Thank
सा कनेक्शन का इशू है ना? ठीक है, आगे बढ़े तो हम लोग.
Excuse me, sir.
बोलो.
Sir, can we by RS ratio can we determine the quality of soil?
Biological quality of soil can be done. Okay, so if you go for the fertility evaluation, there are three properties of the soil fertility: biological fertility, physical fertility, and chemical fertility of the soil. So biological fertility of the soil can be compared on the basis of RS ratio, है ना? अगर दो सॉइल है तो उसमें तुम RS रेशियो के आधार पर उसको कौन सा बायोलॉजिकली एक्टिव है और नॉन एक्टिव है ये बता सकते हो. ठीक है.
Okay, sir.
और कोई?
सर राइजोस्फेरिक इंजीनियरिंग में ओपाइन कॉन्सेप्ट बारे में बोल देंगे.
क्या बोल रहे थे?
सर राइजोस्फेरिक इंजीनियरिंग में ओपाइन कॉन्सेप्ट और एक बार एक्सप्लेन कर देंगे.
ये जो कॉन्सेप्ट है ना, ये सब जो कि बायोटेक्नोलॉजिकल अप्रोच है, समझे? तो इसमें बायोटेक्नोलॉजिकल अप्रोच में क्या करते हैं कि पहले हम राइजोस्फेरिक स्टडी में ये पता करते हैं कि जो राइजोस्फेरिक माइक्रोऑर्गेनिजम्स है वो उसके लिए कौन सा मटेरियल जो कि है फेवरेबल है, एज ए फूड मटेरियल कौन सा फेवरेबल है, उसको किसको फेवर करता है और किसको सबसे पहले यूज करता है. ठीक ना? तो उस तरह का मटेरियल जो कि है सिंथेसाइज करने के लिए हम लोग ये बायोटेक्नोलॉजिकल अप्रोच के थ्रू उस तरह का ये जीन एक्सप्रेशंस को हम लोग डिसाइड करते हैं ताकि जो कि सिंथेसिस ऑफ एक्सुडेट्स जो कि है ऑर्गेनिक मटेरियल जो सिंथेसाइज हो तो कार्बन कंपाउंड्स इस तरह का हो जो कि वो फेवर करे माइक्रोवेल पॉपुलेशन. ठीक है, तो मेनली कॉन्सेप्ट यही है. इसके अलावे जो कि है बहुत सारा जो उसका बार्क हुआ या फिर रूटलेट्स वगैरह जो एज हो जाते हैं और जो डिस्ट्रॉय होते हैं तो उस पे कौन सा माइक्रोऑर्गेनिज्म उस पे ऐड करता है, तो ये सब भी डिसाइड करने के लिए जीन एक्सप्रेशन के थ्रू हम उसको ये करते हैं, उसको मतलब मल्टीप्लाई करके फिर से हम उसको एक्सप्रेस कर सकते हैं. ठीक ना?
ओके सर.
कोई क्वेरी हो? ठीक है, तो आगे बढ़े।
Yes, sir.
तो ये कोरम सेंसिंग, इसको पिछले क्लास में मैंने बताया था। इसको अगर जनरल टर्म अगर दें कोरम सेंसिंग को, तो ये जिस तरह से चैटिंग हम लोग करते हैं ना, वो सेम टाइप का ये है कि माइक्रोब्स आर आल्सो चैटिंग for the sensing, for the interactions with the plant root system. Okay, so in this case, a phenomena whereby the accumulation of signaling molecules enable a single cell to sense the number of bacteria or cell density. है ना? So this one is one of the phenomena where accumulation of signaling molecules, that is the organic materials, organic compounds which are synthesized into the rhizosphere, is enabled to sense the microbial population. Okay, and it will be depend upon the cell density also, and also on the chemical compounds density also. Okay, so the microorganism produce a specific auto-inducer compounds, है ना? कुछ ऐसे कंपाउंड्स सिंथेसाइज करते हैं जिसका कि पॉपुलेशन, जैसे ही पॉपुलेशन इंक्रीज करेगा तो वो जो कि है स्पेसिफिकली जो कि है उसको आइडेंटिफाई कर लेगा. So the microorganisms produce a specific auto-inducer compounds, and as the population increases and the concentration of these compounds reaches critical level, a specific genes are expressed, है ना? तो जैसे ही केमिकल कंपाउंड का क्रिटिकल लेवल इंक्रीज करेगा, माइक्रोबियल पॉपुलेशन का डेंसिटी इंक्रीज करेगा, वैसे ही जीन एक्सप्रेस्ड होगा वो आपका, जो कि ऑटो इंड्यूसर जीन जो कि है एक्सप्रेस होगा, और जो म्यूचुअल इंटरेक्शंस बिटवीन प्लांट रूट्स एंड माइक्रोब्स जो कि है दैट विल बी एक्टिवेटेड. So many bacteria rely on quorum sensing to control the expression of genes responsible for disease, and these are very important for the pathogenic bacteria during infection of a host to coordinate their virulence in order to escape the immune response of the host in order to establish a successful infections with the plant system. So the most common signals in gram-negative bacteria are acyl, homoserine, lactones, and in case of gram-negative bacteria, it is often oligopeptide signaling is there. Okay, so this is all about quorum sensing. Now, the interactions in the rhizosphere. So several type of interactions will takes place in the rhizosphere. As you know, the microbes, a diverse group of microbes are present into the soil, and when seeds are sown, roots are comes out, plants are plant growth takes place. Interactions with the microbes, with the plant systems will takes place. Even interaction between the microbes are also takes place. So these interactions into the rhizosphere are very important. The interactions are both type, both they may be positive, they may be negative, or there may be mutually positive. There may be one positive action will be there. On the another hand, on the another microbes, there may be negative impact will be there. On both type of microbes, there may be negative interactions will be there. So the ultimate impact of these interactions are mainly positive. That's why the plants are survive and several biochemical functions into the soils are going on. So these interactions are very much important. These interactions may be classified into two groups. One is positive, where the overall impact is positive on the interaction, whereas another one is the negative interactions, where the impacts are negative. Okay, so the neutralism, commensalism, symbiosis, protopopulations, these four interactions are well known into the soil, which have certain positive impacts on the interactions, whereas commensalism, competitions, parasitisms, and predations have negative impact. Okay, so the plant bacteria interactions are classified into four categories, eight categories, in which the four, that is neutralism, commensalism, symbiosis, protocooperations are positive interactions, whereas commensalism, competitions, parasitism, and predations is negative interactions. So if two species of microbes and plants are there. One species A is there, a species B is there. If there is no effect on both species, then it is called neutralisms. Commensalisms, when one species have positive impact, whereas other have no any impact, then it is commensalism. Symbiosis, both have positive interactions, but they are not obligatory. Proto-cooperations is positive interactions also, but they are obligatory. Amensalism, one have no impact, but other have negative impact. In competitions, both have negative impact. Parasitism and predations, one have positive effect and another have negative impacts. Okay, so these are 8 interactions which are very important in the rhizosphere. We have to go through each interactions in details also. So neutralisms means there is no impact. Each and every in every environment, if two type of microorganisms are there, there will be no impact on there. So this type of interactions are never found into the systems, and each organisms behave independently, and this kind of interactions probably does not exist in the soil community. Okay, so this type of interaction is never found into the soil system, neutralism. So the organism living in same habitat would have absolutely no effect on their neighbors, है ना? तो अगर दो तरह के ऑर्गेनिजम्स अगर किसी एक सिस्टम में है और उसका एक माइक्रोब्स का दूसरे माइक्रोब्स पे कोई इम्पैक्ट अगर नहीं है, तो उसको जो की है न्यूट्रलिज्म में रखते हैं, बट दिस काइंड ऑफ इंटरेक्शन हैज नेवर फाउंड इनटू द सिस्टम. देन कॉमेंसलिज्म में एक पार्टनर को पॉजिटिव इम्पैक्ट होता है और एक पार्टनर को कोई इम्पैक्ट नहीं पड़ता है, उसका कोई बेनिफिट या ये नहीं है ना. सो दिस टाइप ऑफ इंटरेक्शन आर वेरी मच प्रिवेलेंट इनटू द सॉइल सिस्टम, एंड इन दिस सिस्टम ए पार्टनर डिराइव द बेनिफिट ऑफ द कंपनी ऑफ द एसोसिएट विदाउट अफेक्टिंग इट, है ना. सो ए पार्टनर derives the benefit of the company of the associate without affecting it, है ना. तो one partner is get benefited, where that benefit is derived from the another partner, but that partners have no any impact of the their benefits. Okay, so in this case one organism is able to grow or function as a result of the action of a second organism, है ना. तो एक ऑर्गेनिज्म अगर कोई फंक्शन करता है, उसका जो बाय प्रोडक्ट आता है, दैट इज यूटिलाइज्ड बाय द सेकंड ग्रुप ऑफ ऑर्गेनिजम्स. Okay, so one organism is able to grow or function as the result of the action of a second organism. So, like beneficial association involving two species, one of which can attack a substrate not available to second organism, but the decomposition results in the formation of products that is utilized by the second organism. है ना? Like decomposition of any type of chemical compounds into the soil system, organic compounds. कोई भी organic compounds are acted by one microorganisms, they are converted into the second type of compounds, but after decomposition, that second compound is utilized by the another group of microorganisms. Okay, so it is beneficial association involving two species, one of which can attack a substrate not available to second organisms, but the decomposition results in the formation of products utilized by the second. It is most common microbial interaction in the soil. Okay, so this type of microbial interactions are very much common into the soil system. Example, organic matter decompositions. Like there are several type of microorganisms are there, cellulotic type of fungi, non-cellulotic bacteria and fungi. So cellulotic type of fungi decompose act on cellulotic type of materials, and they are converted into non-cellulotic materials, and then the non-cellulotic bacteria and fungi act on those materials are work as a food material for the non-cellulotic bacteria and fungi. So this type of interaction is very much important. Okay, so two cellulose composers having enzyme called C1 act on undegraded celluloid. Later, partially decomposed cellulose can be metabolized by many soil microbes. है ना? So the true cellulose decomposer microbes have enzymes, and that enzymes, when acted on the cellulose, they are converted in the different components. Okay, so after decompositions, uh, these materials are again metabolized by the another group of microbes. Example 2, that is after rupture of cell wall, protoplasmic materials are metabolized by the other group of microbes. So one microbes acted on the microbes. क्लास के बाद आए हैं थोड़ा सा, हाँ क्लास ले रहे हैं, 15 मिनट में छोड़ देंगे, हाँ आ जाए. After rupture of cell wall, protoplasmic materials are metabolized by other group of microbes. Okay, so cell wall are ruptured by the one group of microbes for their food. The protoplasmic materials which comes out from the cells are acted by the another group of microbes. Okay. Another one is the need of microbes for growth factors, which are synthesized by certain microbes, and their excretion permits the proliferations of nutritionally fastidious soil inhabitants. Okay, so microbes for the growth factors, several microbes synthesize vitamins and hormones into the plants, into the rhizosphere. So these materials, vitamins and hormones, are used by another group of microbes also. Okay, other group of plants also. Okay, so the material which are synthesized by one group of microbes are not utilized by the another group of are utilized by the another group of microbes. Okay, so need of microbes for growth factors which are synthesized by certain microbes, and their excretion permits the proliferations of nutritionally fastidious soil inhabitants. Okay. Another example is obligatory anaerobes. Okay, so obligatory anaerobes. What is obligatory anaerobes?
which can only survive in anaerobic condition.
ये
सर
obligate है ना?
सर जिसको एनारोबिक कंडीशन ही चाहिए वो भी सरवाइव करने के लिए, अगर ऑक्सीजन प्रेजेंट होगा तो उनकी डेथ हो जाएगी.
Yes. So they cannot function in the presence of free oxygen. Okay. So in the soil, co-occurrence of aerobic microbes in the mosaic of aerobics and anaerobic microbes, microsites on the surface of the most soil ecosystem facilitate development of strict anaerobes. So aerobes on the surface of these mosaics or these aggregates, they respire very fast and create microenvironment which are suitable for the obligatory anaerobes, or they create anaerobic environments by the fast respirations. Okay, so like Azotobacter. Azotobacter is one of the microbes which have very fast respiration rate. They require very much organic carbon for their food purpose. But due to respirations, they are developing anaerobic environment in the microatmosphere. And that microatmosphere is utilized by the obligatory anaerobes. Okay, so they cannot function in the presence of free oxygen, but co-occurrence of aerobes in the mosaic of aerobics and anaerobic microsites of the surface of the most soil ecosystem facilitate development of strict anaerobes. Okay, so on soil aggregate surface, aerobes grow and develop environment for anaerobes to grow inside the aggregates. Okay, so organism on the surface did not get benefit from the internal population, because in the internal population anaerobic microbes are there, and suitable environment created by them is on the surface. Okay, so the microbe on the surface create anaerobic environment for the microbes inside the surface, that is anaerobic in nature. Okay, example 5 is relationship between acid sensitive relationship between acid sensitive organisms that grow in soils of low pH only in the immediate vicinity of population that decrease the acidity of the microenvironments inhabited by the two populations. Okay, so the relationship between the acid sensitive microorganisms, जो microorganisms acid sensitive है, मतलब बहुत ज़्यादा acidity डेवलप होने से जो acidity डेवलप होने से आपका सर्वाइवल उसका अफेक्ट होगा. So the relationship between acid sensitive organisms that grow in soils of low pH. Okay, only in the immediate vicinity of the population that decrease the acidity of microenvironment, like the phosphate solubilizing microorganisms, which secrete acids in the reduced substance of the surrounding environment. So in that pH environment, the acid sensitive microorganisms are surviving into the soil. So the phosphate solubilizing have natural property of secreting the organic acid in the surroundings, or they secrete organic acids and create the surrounding environment, although it is microsites which becomes acidic in nature, and that microsites are suitable for survival of acid sensitive organisms. Okay?
Excuse me, sir. It's written there, decrease the acidity. It should be pH or acidity.
एसिडिटी और पीएच दोनों एक ही जैसा करना है. एसिडिटी अगर इंक्रीज करेगा है ना, तो पीएच डिक्रीज करेगा.
So यहाँ पे decrease the acidity लिखा हुआ, vicinity of population that decrease the acidity. So how can the acid sensitive organisms survive?
एसिडिटी डिक्रीज अच्छा इंक्रीज होगा है ना, इंक्रीज एसिडिटी. ठीक है ना, जैसे फॉस्फेट सोलाइजिंग माइक्रोऑर्गेनिजम्स है जो भी बाइफास्फोलिक या सुडोमोनास स्टियाटा या बहुत सारे फंगस है, एस्परजिलस नाइगर है, रिबस है, इसमें जो फंगस है दैट इज सेक्रेटिंग ऑर्गेनिक एसिड. ओके, सो इन दैट एनवायरनमेंट द सेंसिटिव ऑर्गेनिज्म acid sensitive organisms are survive. Okay. Number 6, the decomposition of number of products of pesticide is occasionally more rapid in milk culture than in pure culture. है ना, तो जो भी कंसोर्टिया वगैरह बनाए जाते हैं, जो भी जैसे प्रोडक्ट जो आता है, जो माइक्रोबियल प्रोडक्ट जो आते हैं, तो उसमें मल्टीपल माइक्रोब्स डाले जाते हैं ताकि जो कि है ये रैपिड एक्शन हो, है ना. सो इन द सेम वे डिकम्पोजिशन ऑफ नंबर ऑफ प्रोडक्ट्स and pesticide is occasionally more rapid in mixed culture than in pure culture. तो बहुत सारे कल्चर को अगर मिक्स करके आप देंगे तो जो बाय प्रोडक्ट आएगा that may be metabolized by the large group of microorganisms. That's why the rapid decomposition of the pesticide takes place and toxicity of the product will be minimized. Okay. Now the another one is the protocol basis. Okay, so in which both type of microorganisms, both interactions are relative, both partners are mutually beneficial, but it is not obligated for their existence. So in a medium that is deficient for nicotinic acid and biotin, neither Proteus vulgaris nor Bacillus polymeria multiply, and the former require nicotinic acid and the latter require biotin. है ना, तो अगर अलग अलग मीडियम में हम बोलना चाहें, Proteus vulgaris and Bacillus polymeria, they never grow without the nicotinic acid and biotin, but when we grow them in the same medium without addition of nicotinic acid and biotin, they will grow because the required materials are synthesized by the partners microbes. Okay, so in mixed culture, in the same media, both grows in the partner bacterium synthesize the missing vitamins. Okay, so in a medium which is deficient for nicotinic acid and biotin, neither Proteus vulgaris or Bacillus polymeria will multiply in the separate medium, but in the mixed culture, in the same media, both grow since the partner bacterium synthesize the missing vitamins. In the same way, partners also B vitamins and amino acids are required to stimulate the growth. Thiamine is frequently required by the bacteria. Biotin and vitamin B12 are essential for bacteria and need to supplement in the pure culture. Okay, so the synergism between the vesicular mycorrhiza and rhizobium and legume plants and PSV, Rhizobium and legume plants interactions are example of protogophers. They improve the benefits of the plants. Okay. The symbiosis. Symbiosis, the next most important interactions which are explored very much in the agriculture also. So, an association between two organisms, that is the between plants and microbes, where both the partners derive mutual benefits, and a particular process of nitrogen fixation will be activated only when the interaction symbiosis interactions will take place. So after only symbiotic relationship, the interactions will take place and the nitrogen fixation takes place. Like in the case of beneficial nitrogen fixation, biological nitrogen fixation, rhizobium in legumes, rhizobium survive in soil, but they did not fix nitrogen without their symbiotic relationship of legumes. In the same way, legumes can survive and grow well into the soil, but they cannot fix nitrogen without rhizobium. Okay, so the interaction between rhizobium and legumes, where rhizobium act as a microsymbiont and legumes act as a macrosymbiont, are essential for starting the nitrogen fixation process. So, through the symbiosis process, the nitrogen fixation process is initiated. Otherwise, they can go to the fields separately, but the nitrogen fixation is never takes place. Okay. In the same case, herbaceous microorganisms, more will be there, absorption will be there, takes place. But when the interactions are there with the plant system, another one is the algae and fungi relationship in regions. Okay, so algae benefits by protection that is afforded by the hive that envelop and protect from the environmental stress. Okay, so the algae is benefits by the protection afforded by the hive that envelop and protect from the environmental stress. Another one is the protozoa in underground termites. Okay, so in the termites guts, these protozoa are there, and protozoa converts into logging of the woods. Okay, in the same way, some fungi and ant relationship are there. So the ants which are living into the soil and making a big hole and big, they, they collect leaves as a food material. And in these food materials, they incorporate certain fungi into these leaves, and ant masquerade leaves, flowers, or other plant debris and introduce some specific fungi. They flourish there and provide cheap and possible sources of nutrients after a certain period. So these are the interactions comes under the symbiotic relationship. That is, bacteria resides within protozoa, cells obtain nutrients and protected environments in the protozoa also. Okay, so these are the interactions which takes place into the system, and these are the symbiosis, where the one is the microsymbiont, other is the microsymbiont, and a specific processes are initiated one in the interactions. Then the negative interactions, competition is one of the important interactions in which, for example, if you add some organic material into the fields, then a group of microorganisms populations are comes out. Okay, so the population will grow very fast. There will be competitions for the nutrients. Okay, so the universal phenomena of coexistence in a community where one species is suppressed as the two species struggle for the limited supply of a nutrient or other common requirements. Okay, so when the food requirements, foods are limited, then a good competitions will be there, and the species are the struggle for the limited supply of food and survive. Only those microbes will survive there, which are have a diverse group of source of food material that can survive on the diverse type of substrate material. So addition of substrate material in the field increasing microbe population will be there is an example of a competition. Then amensalism. So amensalism is a kind of relationship in which a partner suppresses the growth of other partner by producing toxins like antibiotics, harmful gaseous compounds like ammonia, carbon dioxide, ethylene, sulfur compounds, nitrous oxide, H2O2, hydrocyanide, etc. So, in this case, commensalism, a group of one species can uh, suppress the growth of other partners, other group of microorganisms by producing certain type of substances, certain type of chemical compounds, certain type of antibiotics. And in the case of antibiotics, only a small quantity of antibiotic is sufficient for suppressing the growth. Whereas for the gaseous compounds or toxic materials, a certain concentration of these materials are essential for the suppression of the growth of the other group of microorganisms. Okay, so sometimes the growth of some fungi may be affected adversely by the large amount of ammonia released during decomposition of manures. Okay, so this is a common example in which the green manuring are done. Green manuring, that is, the leguminous plants are incorporated into the fields and decomposition takes place. So during the decomposition, very rapid decomposition takes place and nitrification will be also takes place. Ammonia involved in very large amount. Okay, so the higher quantity of ammonia suppresses the growth of the nitrobacter also. Nitrobacter is a type of microbes which are involved in the nitrification, conversion of ammonia into the nitrate. Okay, so sometimes the growth of nitrobacter and some fungi may be affected adversely by the large amount of ammonia released during the decomposition of leguminous green manures. Like in the case of high-vis H2S that is oxidized and it is converted into the H2SO4 and the pH will be drastically decreased. Okay, so the thiobacillus species mainly takes place into the cat soil or into the acid sulfate soil. Okay, where the H2S are mainly oxidized and then converted into H2SO4 and due to sulfuric acid pH will be goes very high acid conditions will prevail. And this oxidation will take place to the thiobacillus oxidation, thiobacillus bacteria. Okay, so the pH sensitive microbes is inhibited. Okay, after oxidation of H2S to H2SO4, the microbes which are sensitive to the pH, their growth will be affected. Okay, and after oxidation, the pH will drastically decrease or drastically increase the acidity will be there. So the pH sensitive microbes are inhibited. Other examples are biological inhibitors of toxins are produced effective at higher concentration in the case of organic acids, insulators, inhibitory at low concentration, antibiotics in case of antibiotics, like Ascomyces, Oligocenerus, they produce heliomycin also, so antagonistic to the Acinetobacter and Acinetobacter crystalloproteases in sterile conditions or in laboratory conditions. In the same way, Bacillus Penicillium nigricans, they protect onion from the infections with the Sclerotium pseudomonas, the onion white rot disease also. Okay. Like critical populations of oxygens are also the example of emissions leak. So competition for nutrients, especially for the carbon, is assumed to be responsible for the well-known phenomena of hepatitis, that is characterized by the emission of the fungal spores germinating in the nutrient source. Okay. So competition for trace elements also like iron, copper, helium, and magnesium, etc. Like you know about the cytopores. Some microbes are able to promoting bacteria are able to assimilate iron, or they convert iron into organic form, or they chelate, and that makes the microorganisms. Okay, so this type of competitions. For example, iron is essential growth elements for the all living organisms and for the plants also. And the study of its bioavailability from soil has resulted in the previous competitions also. Okay, so they affect the growth of the microorganisms. So cytophores that is low molecular weight compounds. Okay, with high iron affinity, usually biocontrollers of plant growth bacteria, soluble iron, and completely acquire iron, not the iron exists, and that by any iron unavailable to other soil microorganisms that don't grow like of it. And these type of microorganisms are typically the pathogenic microorganisms or deleterious type of microorganisms. Okay, so they affect the growth of the pathogens and deleterious type of microorganisms, but biocontrol agents and the BPR are fast. The bioavailability of these irons are suitable for the plants only. Originally synthesized the cytophores, except the iron cytophore complex by using a structure that is specific to the complex located in the outer cell membrane. Okay, then parasitism. So it is a situation that one partner lies in or on the body of the other and feed on the body fluid of the host. So it is a host-parasite relationship in which one is benefited while other is adversely affected. Okay, so host-parasite relationship in which host is benefited, whereas host-parasite relationship in which one is benefited, other is adversely affected, like earthworm bacteria relationship, like actinomycetes, where they are feed on these microbes. The biocontrol agents for control of root rots and wilt disease, they control the disease by the these rules. Then the predation. Predations, it is the direct feeding of one organism on the others. So highly prevalent in case of protozoa, they feed on the bacteria to strike a balance between the population of prey and predators. I have given you example in the case of the rhizosphere populations. If you found the protozoan activity, it means there are heightening of the microbial population, bacterial population. Like in the case of mixed molds feed on the soil. Okay, that's all for the interaction, microbial interactions, and I have told you that all these interactions are going into the soil. Always interactions are prevailing into the soil, but the ultimate, although some have beneficial effects, some have negative effects, but the ultimate, the overall impact of these interactions are positive, and for that, there will be existence of the plant system or several biofunctions into the soils are going on, and the nutrient release pattern, enzymatic activity, good microbial activities, and nutrient release into the soil after a decomposition are there. Okay, so that's all. We will meet in the next class on Wednesday. It is. So Wednesday is Foundation Day. So that class will be, I think, that will not.
Excuse me, sir. Sir, is cytophore microorganism help in decreasing the acidity of the soil, which is caused by
cytophore microorganism काम करता है. साइड्रोफोर्स का काम केवल होता है सर्टेन जो मेटल्स होता है उसको जो है ये चिलेट कर देता है, मतलब उसको बायोअवेलेबिलिटी को अदर माइक्रोब्स के लिए कम करता है ना, लाइक पैथोजेनिक माइक्रोब्स, लाइक द ये डेलिटेरियस टाइप ऑफ माइक्रोब्स है ना, क्योंकि सॉइल में बहुत तरह के माइक्रोब्स है, बट व्हेन यू आर यूजिंग द प्लांट ग्रोथ प्रमोटिंग राइजोबैक्टीरिया और इफ यू आर यूजिंग बायोकंट्रोल एजेंट्स, दे हैव प्रॉपर्टी टू obtain iron which are present into the soil, है ना, so they for the time being they make unavailable for the pathogens and deleterious type of microorganisms. So the growth of these microorganisms are affected, and the growth of the biocontrol agents or PGPR are accelerated or proliferated. They multiply very rapidly into the rhizosphere. Okay, but when plants require iron, they supply these iron. Okay, so they are multiplying to the rhizosphere. Okay.
Okay, sir.
Okay, so that's all. We will meet in the next class. Okay. Thank you very much.
Thank you, sir.
Thank you.